Yeah, this is a great question. And I'm going to take just a couple of minutes on this because I think this is of paramount importance. Um, Number one, these vaccines are the culmination of about 25 years worth of work. So we've really been trying to fight against this message that they're too new. I mean, a quarter century, I guess that's new. And as I get older, I'll probably think 25 years isn't that bad, but it's been a long time. We've known for at least 20, 25 years that that spike protein is the important thing we need to target. That's number one. Number two is people have been working on mRNA technologies for either gene therapy or other vaccines for 2 decades. And those were going along, and then they came together during this Operation Warp Speed when we tried to respond to the pandemic. Second thing I would say is that we did not rush the vaccine trials. I think Operation Warp Speed and the prior administration deserves a lot of credit for setting us up for success. It was almost a business model of saying, what can you do, Manhattan Project style, if you have the funding needed, you have the commitment needed, and everybody's doing the same thing. You mentioned it in the intro. I work on flu and staph and other vaccines. Not right now. Everything is COVID. We eat, sleep, and then COVID. Like it's what we do. And that's what our HIV colleagues are doing. And that's what our flu people are doing. It's all about COVID right now. The ability to do that made this go quickly. So we didn't rush them. We just did them in a way that government is not typically known for. It's being efficient and effective. And we were able to do that. Third thing I would say is we went through the necessary steps of phase 1 testing, first in people, phase 2, where we tweak the doses a little bit, phase 3, where we prove in tens of thousands of people that it works and that it's safe. And that gave us the information we needed. And we did those so quickly because we were already writing the studies and the protocols even during those early days of February and March when we weren't even sure if a vaccine was going to be available. We went at risk and said, let's go ahead and write the protocols as if we're going to be successful. So that's number one. Number two, we took advantage of the emergency use authorization mechanism. Usually vaccines need about 6 months of follow-up data before they can be licensed. In this case, we allowed for 2 months out of necessity. The reason we did that is that when you have side effects from a vaccine, they always start in those first 6 to 8 weeks after vaccine. And that doesn't mean you know everything about them in that time period, but if someone makes it through 2 months and there's no new onset of something, we can really reliably say that it's uncommon and unlikely that that has any relationship to the vaccine whatsoever. Third is we understood the biology. We used this in different animals. We gave animals that were pregnant or animals that were newborns very high doses of vaccine to make sure it was safe before we ever put it into humans. And then the last thing is we've asked many hundreds of thousands of people, it's in the millions now, to fill out diaries after each of their vaccines on a special app to be able to track those symptoms along. And by doing that, we've been able to find some rare adverse events or side effects after vaccines, like heart inflammation, about 1 in a million, blood clots after Johnson & Johnson, about 1 in a million. We saw allergies early on, but we haven't seen those types of adverse events that maybe people were worried about, like changes in fertility or other unusual issues. Last thing I would say about this, and I think this is really important, these mRNA vaccines are really the most trimmed down, svelte version of a vaccine that we have seen in a long time. It's really just 4 ingredients. It's the mRNA, which is just the genetic recipe for that spike protein. We've got to protect it because your body gets rid of it really quickly. So we put it in a fat bubble or a lipid nanoparticle. There's a little bit of sugar and salt just to balance things out and a buffer to make sure that it's not too acidic or basic. And that's it. It's not grown in a chicken egg. It's not done. We don't do anything else with it. So because of that, there's really not much in that vaccine that's going to cause problems. That gives us a lot of encouragement as we do the clinical trials. So what have I been doing for the last 6 months? We've been vaccinating children down to 6 months of age in very carefully controlled clinical trials so that we can document ad nauseum every single side effect that they might have: fever, leg pain, achiness after vaccine, as well as those more potentially significant side effects, which we thankfully have not been seen. So I can tell you that as the parent of 3 children, one of whom is a 4th grader who had disease with us back in March 2020, She's gotten her first dose of vaccine, so she's counted in that 9%. And she was pretty excited to be able to do that.